Hello, this is Jesus Avalos. I am the Biotech 2 course lead for the 2025-2026 academic school year for IDEA Public Schools. Today we are going to dive into Unit 6 internalization tips that could help out teachers across the biomedical science program. Some of the objectives are the following. Welcome, objectives. Number 2, unit overview. Number 3, big questions and standards. Number 4, weeks at glance. Number 5, planning and resources and next steps. So, Unit 6, what is it composed of? What is the overview? The unit length is about 13 days. The scanning deadline will be January 29, 2026. What is the main focus of this unit? DNA extraction and PCR amplification for barcoding, the use of sequencing data for serious species identification, application of bioinformatics databases and alignment tools, project-based work, conducting a DNA barcoding experiment and presenting results. What are some of the outcomes of this Unit 6 overview? The unit emphasizes in DNA barcoding, bioinformatics tools, and experimental design. Students will connect sequencing concepts to biodiversity research and real-world applications. So, what are some of the big questions and standards over this whole unit as a whole? Number 1, how does DNA barcoding help us identify organisms and understand biodiversity? Number 2, what tools do scientists use to analyze DNA sequences in bioinformatics? Number 3, how do experimental design and accuracy ensure valid results in biotechnology research? Now, what TEKS does it tackle? Tackles TEKS 6AB, 10A, and 11B. What are some of the key understandings of this unit? Number 1, DNA barcoding links molecular techniques to ecology and medicine. Number 2, bioinformatics tools, for example, the BLAST, are essential for analyzing sequencing results. Number 3, accurate experimental design supports reliable scientific conclusions. So the week 1 at glance, this unit is pretty short. So day 1, introduction to DNA barcoding and its applications. Day 2, DNA extraction and PCR amplification. Day 3 through 4, bioinformatics intro using BLAST/GenBank. Day 5 to 6, DNA barcoding, project lab species identification, and day 7, careers in biodiversity and genomics. Week 2 at glance, day number 8, data analysis of sequencing results. Day number 9, project refinement. Success day, practice presentations. Flex day, lab or analysis extensions. And the unit exam day, final project presentations. So what are some high leverage planning tools or tips for this unit? Number 1, ensure PCR machines, primers, and reagents are always ready. Number 2, confirm access to NCBI tools and computers. Number 3, prep unknown DNA samples for student barcoding projects. And last but not least, use vocab and discussion stems early and very often. What are some must-use resources? Slide decks, linked in roadmap. Prep rubrics aligned to standards. For example, data accuracy and communication. Wet lab YAG for barcoding, and last but not least, DNA barcoding protocols, which can all be found in the wet lab YAG. So what are some final thoughts plus next steps? Scaffold student use of bioinformatics tools. Number 2, reinforce professionalism in scientific presentations. Number 3, prep rubrics aligned to standards, data accuracy, and communication. And last but not least, number 4, use the tips library for ongoing support. Thank you. And as always, if you have any questions, my email is linked right on the slide deck. And assigned is the link to all Unit 5 resources. Thank you so much for your time and for taking a moment. Hello, I am Jesus Avalos, the Biotechnology 2 course lead for the 2025-2026 academic school year for IDEA Public Schools. And today we're going to dive into Unit 7 internalization tips. So, let's get started. Objectives for the slide deck. We have number 2, the unit overview. Number 3, big questions and standards. Number 4, the weeks at glance. Number 5, planning, resources, and next steps. So the unit overview. It is approximately 9 days, so a very short unit. The scanning deadline is February 12, 2026. The focus: expressed gene insert into host cells, extracted, cell lysis and inhibitor protection, quantified UV/vis spectrometry, MBCA assay, Separate SDS page, electrophoresis, visualize como se and code blue staining. So what are the outcomes? Unit emphasizes real-world lab technique, experimental accuracy, and data interpretation, key skills used in biopharma, diagnostics, and academic research. So very, very heavy unit regarding to research. So what are some of the big questions and standards for this unit that teachers should be asking and provoking to the students? How is protein expression carried out in yeast stems? What steps are required to extract and quantify proteins accurately? How do SDS page and staining techniques allow scientists to analyze protein composition? What teaks will be targeted? 12A, 12B, 12C, 14B, 14D, 11C, and 1B. Some key understandings for this. Yeast can express eukaryotic proteins because it performs post-translational modifications similar to human cells. Number 2, protein extraction requires careful lysis and inhibition to protect protein integrity. And number 3, SDS page separates proteins by molecular weight to allow comparison and identification. So the week at glance. Day 1, Intro to Protein Expression Analysis Workflow. Day 2, Protein Extraction Lab Part 1. Day 3, Protein Quantification Lab Part 2. Day 4, SDS Page Lab Part 3. Day 5, Gel Staining and Band Analysis. Day 4. Part 4. Week 2 at glance. Flex Day, Data Analysis and Sequence Results. Success Day, Unit Review / Protein Analysis Reflection. And last but not least, we have the exam day. So what are some high leverage and planning tips? Number 1, limit lab groups to no more than 6 per class. Resources are pretty tight for this unit. Number 2, prepare YPD media, YPER buffer, protease inhibitors, and BCA assay reagents ahead of time. This will always help you out. It's always to be prepared than to be underprepared. Number 3, ensure access to spectrometers, SDS page systems, and power supplies, and code blue stains. Number 4, students must take photos of gels for analysis. So what are some must-use resources? The slide text, as always, mentioned and linked in roadmap. Gel electrophoresis and staining protocols, which are very easy to understand and broken down. The wet lab yak is always going to be such a great resource for any kind of lab and to find any kind of protocol. And as always, there has been an exam review teacher guide provided as well in the hub. So what are some final thoughts and next steps? Number 1, review common errors from SDS page and BCA analysis to reinforce accuracy and scientific reasoning before advancing. Number 2, maintain lab equipment by cleaning electrophoresis chambers, disposing of gels properly, and restocking protease inhibitors and buffer reagents. Number 3, begin sequencing guest scientists, community partners, or volunteer judges now. Why? Unit A requires outside feedback. Okay, keep that in mind. It's better to, as always, be prepared than underprepared. Number 4, use the tips library for ongoing support. Thank you. If you have any questions, my email is allocated on the slide deck. And as always, guys, the link will always be provided to you on the slide deck as well. Thank you for taking your time to dive into the